Hello. Yeah. So how are things going with you?
Well, I got I got onto Chat GPT and I decided they're my new help desk. They told me how to get a stress strain curve out of Console.
Oh, good.
Console didn't I checked online and ResearchGate and their help desk and the help desk through the CMC thing that I'm with. They didn't seem to understand what I was asking.
Huh.
You know how to do it?
Yeah.
They put a probe in.
Yeah.
They have probes, so I put 5 in.
Wow. Yeah.
Way more than I need.
Oh, so yeah.
Remember that if you're working with Console, you put a probe in, and you can put a point probe into it, or a line probe, or just wherever you want in the model. You just go click on a node and you say, "Oh, I want the probe over here," and now it's actually generating data for me.
Oh, okay.
And with the spring, with a spring tension element, it gives me a sort of a J curve. And with a straight element, it tends to give me a straight curve. So I've got to find something that'll give me a J curve to see if it'll give me a J output. So we'll see.
Yeah. Well, that sounds great. Yeah.
At least, yeah. Well, it's a graph. You click on there and you put it into an Excel file, and then you can do whatever you want with it. You can put it in MATLAB. Oh, it's just great. I knew what to do with it once it generated it. But,
yeah.
well, so remember that Chat GPT is a good help desk.
Oh yeah. Yeah.
Especially for programming.
So that's great. Well, thanks. Yeah, that was. It sounds like you're making progress then.
Yeah, finally. Well, this has held me up at least 6 weeks.
Wow.
Yep.
Yeah. Hello, Jiahang and Sushmanth.
Oh, hi.
Hi, how are you?
Yeah, fine, thanks. And you?
Pretty good.
Yeah, long time no see.
Yeah, yeah. How are things going for you?
Yeah, all good, I think.
Yeah. Yeah, well, so we're getting ready for GSOC. A couple more weeks. I think next week maybe, or the week after they announce. So right now I think we need to get things ready. Just, if we have things in the repository, I think I mentioned this, that we want to clarify or make sure that they're in place. We should do that now. I also have to put together a guide. I haven't done it yet, but things that we need to clarify about some of the data. But that's on me, so I'll have to do that. Yeah.
Yeah. Actually, I'm for now maybe you remember I have mentioned one paper maybe in the last year, the GNN for cell tracking. There was one paper which has been published in the ECCV 2022.
Yeah.
And actually, in these few weeks, I'm working on reproducing the results of this paper, and I think this model is a good, I think, starting point for our project. But for now, my work is not done yet. I'm still working on it, and there are some problems with the source code, so maybe not very quick. And for the GSOC projects, because maybe I have missed much progress regarding the GSOC. Yeah, so I think I need some time to like catch up with the progress of other people. And yeah, also I'm going to prepare the DevoGraph, the project, so that other participants can contribute to this project. Yeah.
Okay.
Yeah.
Yeah, in terms of the DevoGraph specifically, people really haven't done much with that, but Sushmanth has been working on like upgrading DevoLearn, which is the sort of the segmentation and tracking aspect. So the segmentation part is he's really been doing some good work on that, and then we're also getting things up onto Hugging Face. So that's like an open model repository. It's not just GitHub, but it has like we can host things like documentation and so forth there. So
yeah. I saw those Hugging Face stuff. Yeah, but I didn't try them yet. Yeah, I will take a look. Yeah, later. I think.
Okay. Yeah. Mm-hmm. So yeah, I don't know how we're gonna proceed with GSOC. I don't know how many people will get. I requested at least 2 people, so I don't know.
Yeah.
Hello, Sushmanth. Thanks for the update, Jiahang.
Kind of traveling. I am kind of traveling right now. If my sound is like disturbed or something, like please understand it. I'm in kind of middle of traveling. I'm coming from my home to college, and last week I haven't done lot of work actually because my brother met with an accident and he got with a surgery. So I was caught up with that, but so I couldn't done a lot of work. And right now I see the segmentation mode for DevoLearn. The stage one of DevoGraph was extracting the centroids. Some part of video frames in DevoGraph are not extracted properly. That's what I want to upgrade it. So I was using instant segmentation model. I want to develop an instant segmentation model for C elegans using PyTorch library and image scikit image library. I have started writing code actually. Right now working on data augmentation part. And I was contributing to DevoLearn DevoWorm website also. And I was working on Hugging Face Devo model of API. I want to host a complete DevoWorm I was working on it parallelly. And that's what my whole week went on.
Okay. You're breaking up. Yeah, I think you broke up there. Yeah, I heard you're talking about data augmentation and the scikit library and the website work. Now the website work, yeah, I saw a lot of the push. Okay, yeah, that's good. I saw a lot of the pull request stuff. Okay, half an hour, and that looked good. I keep up the good work. I have. It looks like good things are happening there. So, for I think last week we talked about how we have the DevoWorm.github.io, which is a place where we usually have various like tutorials and things to go over. And they've been Sushmanth and Jyothi, another contributor, have been updating that quite a bit. They've been putting in pull requests. It just needed an update. It was aging, so it's been a couple of years since we've touched it, but it looks like we're getting some pretty good things up there. So, we'll be wanting to, of course, generate documentation along with code during GSOC, so that'll be a good opportunity to host that somewhere. We have also blog, so we have the blogs from past GSOC projects. Did you do any blog? I don't think you did any blogging, Jiahang. last year.
Yeah, I didn't write any blog.
Okay, yeah, but you did do some work on like documenting sort of plans for things too.
You mean documentation of DevoGraph?
Well, like you had some presentations that we went over last year.
Yeah, but I didn't record those presentation, I think.
Oh, well, I mean, yeah. So I don't know what we want to do in terms of documentation for DevoGraph, but if you have some things that you don't have in the README in the repository, yeah, you might
Yeah.
we might do that as well. Put that in a basically we have these blog posts that have been put up here, but I'm thinking that we use that as like a documentation um, aspect to the project, so people can go through those and read more about what was done, or things that for the future or whatever.
Yeah. I got your point. Yeah. Actually, because of DevoGraph, for now, the status of DevoGraph is just I think it's not very mature. Like we need to do a lot of things to like finish this project. So I didn't prepare a full document for the DevoGraph, but I think this is one of our jobs in this summer, the GSOT 2023. We want to do more on the DevoGraph, including the documentation part. So yes, and furthermore, I think if we have successfully finished the DevoGraph with great documentation, and we can put some descriptions or introductions of DevoGraph in the blog post, I think.
Yeah, I think so. That would be good. All right. So yeah, we'll go. We'll kind of target that for like a thing to do for the summer, and we'll also see what who's participating this summer, and so that should be good. It should be a really good time, and we'll do. We'll keep our weekly meeting time as a place for showing off work. People can present or whatever they want, and then we might have meetings within the week to talk about specific things that people are problems people are having or anything else, and then we'll use the Slack and the GitHub repositories too. So we'll have that as like kind of a way to communicate with people, and make sure that we can get everything going on time. Hello, Jesse. So that's good. Yeah, so I look forward to this summer. It should be good. Like I said, the community period starts in about a week and a half. So right at the beginning of May. So it's coming up. So, Jesse, how are you? Okay, good. All right, so why don't I get into what we were going to do today? Okay, yeah, he's just listening today. Okay, for right now, anyways. So, we have a new publication. Jesse, of course, and Dick Gordon and myself. Let me share my screen. So this is something that's been around. We've talked about it for a while. There's actually a presentation on the YouTube channel on this work. This is on the diatoms work, and this is called The Psychophysical World of Motile Diatom, the Motile Diatom Basilaria Paradoxa. And so, this talks about some of the basilaria work we've done, but instead of being driven by, the last paper we did was a couple years ago, where we did a lot of deep learning analysis of the microscopy data, and then we did some biomechanics analyses. So we looked at the movement from two different perspectives and tried to characterize the morphology of the movement. In this paper, in this book chapter, we talk about what we call the psychophysics, which is like the the response to stimuli that diatoms have, and especially this genus Basilaria, which of course is this colony of cells that moves back and forth like an accordion. And so it has this collective behavior, this movement that's very intriguing. And so we characterized this in the last paper. In this paper, we consider some models and some ideas of how this movement is generated. So this paper kind of goes over a lot of different models of psychophysics that might be applicable here. So there's a lot of good stuff in here about how this organism without a brain is generating this response to stimuli. And so this is something that is we have an open source version of it. It's off of the website, off of publications page, so you'll be able to find it there. This is part of a larger unit called the Mathematical Biology of Diatoms. So this is chapter 9, as you see here. And then this is the book here, which is the Wiley Online Library. So, people may or may not be able to access it. I wasn't able to access it for some reason, but basically it's a whole book of papers on this topic of the mathematical biology of diatoms. So there are a number of different papers here by different contributors. Part 1 is diatom form and size dynamics. So this is really kind of getting into a lot of the diatoms are really fascinating group of organisms because they have their algae, so they have like a solid cell wall. It's actually silicates, made of silicates, and they accrete their cells. They don't necessarily grow them like an animal. And they can divide, and they actually have sexual reproduction, and they respond to different cues in the water column in different ways. So without going into the details of diatom biology, which we have in that YouTube video on the YouTube channel, we can do a number of different things to look at, how they behave, their biology, how they persist over time. So part 1 is diatom form and size dynamics. This talks about size regulation in diatoms and, kind of getting a sense of what the size and shape of diatoms are. So diatoms are very small. A lot of them are less than 100 microns in size, so they're very small. But they are very, the cells themselves are actually quite exquisite. They can form all sorts of shapes. So this is modeling, size resolved modeling of diatom populations on the mathematical description of diatom algae. So they can, the silicaceous exoskeleton structure and properties to colonial growth kinetics and prospective nanoengineering applications. So they use diatoms in biofuel production and in other nanoengineering applications. As you can imagine, you have these very small organisms that are motile, that have silicate bodies, cell walls, and they can move through the water column quite effectively. So you could use this for a number of applications in nanoengineering. But you have to do this, you have to characterize the cells and the different cell types, of which there are many amongst diatoms. And so part 2 is diatom development, growth, and metabolism. So whereas the last section was about just a descriptive aspect of diatoms and using ion beam imaging and or ion beam technologies and some other technologies to measure those. This talks about growth and metabolism. So this talks somewhat about evolution and growth modeling and some aspects of noisy data and characterizing growth and metabolic modeling, which is I think we've talked about metabolic modeling in previous meetings, but it's actually quite, it's quite a tough modeling exercise, but they do this in diatoms here. Then there's diatom motility, and that's where we've come in. Our chapter is in here with a couple of chapters from Thomas Harbeck, who's been in the group before. He did one paper modeling another basilaria species as a chromoto oscillator with time delay. So he gets into the specifics of some of the movement models. The chromoto model is a dynamical systems model. It deals with like excitable oscillators. So it's the original way use it in neuroscience to model neurons and neuronal networks where you have a bunch of neurons coupled. And so he's using that in this case. And then this paper is on Diatoma vulgaris colonies, and this is describing this using a Lindemeyer system. So a Lindemeyer system is a system of growth where you put together different shapes and symbols, and you can grow systems from there. So that's another interesting paper. And then Richard Gordon with Krishna Katyal and Shruti Rajvansh Singh, who have been in this group before. All three of them have been in this group. They did a paper on Rafe, which is simulation of the dynamics of diatom motility at the molecular level. The domino effect hydration model with concerted diffusion. So this is their model of diatom movement. So all four of these papers are really interesting. I would read all of them if you have time, if you can get access. Then there's diatom ecological and environmental analyses. So these are, again, they're mathematical models describing the interaction of diatoms with light. There's this another paper on light response, another paper on like chemical sensing. So this is this diatom species as a biogenic charge-based sensor for heavy metal ion contamination detection. So this diatom can sense, chemical pollution, and it can, respond accordingly, and you can use that as a determinator of whether you have a large amount of this pollutant in your local area that you're testing. So those are the papers in this special issue. I think that'll be, something that people might want to check out. Let us know if you need access to things. We may or may not be able to get them for you. So anyways, congratulations to everyone involved in that. So it looks like we have Himanshu with us. Hello, Himanshu. How are you? And Jyothi's here. Hello. And Sushmanth's back. Do you want to say something, Jyothi? Okay. All right, so that's great. Nice turnout here. Thank you for attending. The next thing I'll talk about is this thing on synthetic morphology. So this is something that we talk about a fair amount in the group about morphology, and the last week we talked about the special issue on symmetries and morphogenetics and things like that. This, we've also talked about this in the context of artificial life and morphological computation. So like you have the body of the embryo, the body of the worm, body of different organisms, and how you can how development shapes that body, what it looks like. We call that morphogenesis. And so this is a Scientific American article on synthetic morphology. And this is synthetic morphology lets scientists create new life forms. So this is a new field that's emerging, and this is sort of based on some of the work that's been done in A-Life. And there's an area called morphological computation, which people have done largely with robots, where they've been able to take a morphology and use different ways of calculating sort of the optimal morphology and different things like that, evolving shapes using genetic algorithms. This is kind of moving this towards the biological realm. So, so they kind of go through some interesting things here at the beginning. They talk about mythical hybrid beasts that you find in mythology, like mermaids, centaurs, and chimeras, which are just kind of like speak to our fascination with biological forms that are different from what exists in nature. And so this kind of leads us into the science of this, where we're going to create like these we're going to create these morphologies that don't exist in nature. Let's see. But a crude stitching of components will not produce a viable organism. Bodies aren't a collection of arbitrary pieces. A human embryo grows into a being with the standard features of a human body, all the parts working in synchrony. Biological forms seem to have inevitable, unique target structures. An emerging discipline called synthetic morphology is now questioning that notion. It asks how and how far the natural shapes and compositions of living matter can be altered. The goal is not to create grotesque creatures such as the Fiji mermaid, but to understand more about the rules of natural morphologies, or more natural morphogenesis, the development of biological form, and to make useful structures and devices by engineering living tissue for applications in medicine, robotics, and beyond. So this is really about like understanding the rules of morphogenesis and how to use that to build these engineered structures. So say you wanted to build an embodied robot, which people often, just kind of put together a body. They say, well, this robot needs a body, or, if we have something like a Roomba robot that moves across your floor to vacuum, it has a body. It can use that body to sense the corners of the room, to sense where it is in the room. But, we can actually do much more with that body. And to do much more with that body, we have to understand the rules by which that body exists in an intelligent agent. So, synthetic morphology might be considered the next stage of synthetic biology. The latter discipline has racked up impressive achievements in retooling cells for non-natural tasks. For example, programming bacteria to glow in the presence of pollutants and other chemicals. Much of synthetic biology involves genetic engineering to introduce networks of genes that give cells new functions, such as manufacturing enzymes to make a non-natural molecule. So this is kind of what we're talking about with diatoms, except people actually create, they take things like bacterial cells. They'll manipulate the genome so they can get only like the essential genome, and then put in specialized genes that will produce things like enzymes and do this efficiently. So you can do the same thing to build other types of organisms, maybe organisms like a diatom with a body, and you can specialize that genome as well. Although I know of no work happening in diatoms or anything really beyond bacterial genomes. So synthetic morphology works at the next level, controlling the shapes and forms into which many cells will assemble. Using the cells of multicellular organisms like us, the technology might allow scientists to design entirely new tissues, organs, bodies, and even organisms by exploiting the tremendous versatility and plasticity of form and function that seem to reside in living matter. And so they kind of go through this. They interview Michael Levin, also Roger Cam, who's does a lot of stuff in synthetic biology. And so then they get into the rules of living form. And this is something that we've been looking for many years, ever since Darwin, in fact. In the mid-1800s, Thomas Huxley began to suspect that there was a generic form of living matter, often called protoplasm, from which the most primitive life forms are fashioned. So this was something that was an idea that, there was a sort of force of morphogenesis. And of course, that was incorrect, but we're getting to, I think, understand maybe what these things are. In his 1912 book, The Mechanistic Conception of Life, German physiologist Jacques Loeb argued that life could and should be understood according to engineering principles. After discovering that he could stimulate asexual reproduction by treating unfertilized sea urchin eggs with simple salt solutions, he became convinced that nature's way of doing things with living matter is not the only way. The idea is now hovering before me, he wrote, that man himself could act as creator even in living nature, forming it eventually according to his will. So this is a very early 20th century quote. It doesn't give specifics, but it's a starting point. And then people figured out how to do tissue culture and things like that. But now we can use tissue culture to sort of serve as the experimental test bed for some of these ideas. The ideas of cells, such as building blocks in our bodies, might make them seem rather passive, like mere bricks to be stacked in the masonry of tissues. So what they mean by cells as building blocks is that cells form larger structures. They form tissues, they form networks, like we talk about with our network, things that we talk about with networks and other things. So cells can be part of larger systems, networks, structural integrity, and organs. And so cells are much smarter than just simple bricks in a wall. Each cell in many respects is a living entity in its own right, able to reproduce, make decisions, and respond to its environment. So this is where they're kind of going with this. They're talking about, using the cells as like sort of components in a system, but it's not like, just putting together like a set of bricks. You have to account for the fact that each cell makes its own decisions, sometimes collectively with other cells, sometimes independently. So then they talk a little bit about morphogenesis. How does the featureless blob of cells, that is the early embryo, know what to make and where to make it? And we talk about that a lot in this group. And we talk about how some of that is, a lot of it is encoded in the genes, some of it is mechanical, and there's this aspect of morphogenesis of sort of this collective behavior, both mechanical and signal, communicative. There are these signaling pathways. Some of them are mediated by genes and some are mediated just by the chemical signals in their sort of spatial location. So there's a lot here. They talk about, they give some examples of cell communication, chemical signaling, mechanical signaling, and electrical signaling, which is also a thing that we kind of think of as something that you see in neurons, but they're actually in all cells. They show a neuron here, but they could just as easily show a skin cell because skin cells also have electrical potentials. So you have these three different channels of communication: chemical, mechanical, and electrical. And so they kind of go through this, some of the aspects of this. They go through this idea of a control knob. So, we can modify some of these communication channels, turn them up or down as desired, and we can build things with cells so that the way they behave collectively is tunable. And you can do this in different ways. They highlight some of Michael Levin's work in the lab, some of the people he works with. And so they kind of get into some of these things about morphogenesis and programmable morphogenesis. Then they talk about the plasticity of cells, we talk about that a lot in the group, which with respect to like stem cells and cell differentiation. And then they talk about reprogramming cells here, which we haven't, I think we've talked about it a little bit in the group, but so cells are also can be reprogrammed in terms of their fate, but also in terms of their shape, which they don't show here too much. They show cells of different shapes down here. So it's important to know that cells have vastly different shapes depending on their type of cell. But cells also, they shapeshift during their existence. So in a lot of the work we've talked about with mechanical stimuli, cells change their shape depending on where they are in a group of cells or what mechanical stimuli they're exposed to. So, cell shape is important as well. And then there's this aspect of programming the shape, which is just basically controlling that process and knowing what it is exactly that's controlling it. Then finally, they talk about this idea of a chimera, which are two different organisms contributing to the same sort of hybrid organism. So these chimeras are very common in mythology. They talk about two systems that are like chimeras. They talk about organoids, which we've talked about before, and xenobots. And so organoids are where you have a bunch of stem cells, you grow them in a 3D scaffold, and you can grow them in different shapes and three-dimensional volumes. Sometimes they'll differentiate into different tissue types, and sometimes they form systems. So they'll form systems of different cell types. Like, you can make a small gut, or you can make a small mini brain, or something like that. This is done in culture, it's not done in the organism. And it's highly controllable. You can measure its activity, you can affect its form and shape. And then xenobots are these, they're based on frog embryos, but they're these little robots that you can build from, based on developmental principles from these little proto embryos, and they can move around and do things in a way that are programmable. So this is really interesting work. They show some different synthetic morphology engineering applications, such as construct, coalesce, develop, and generate. So you can take single cells and you can, put them together in different ways according basically functions. So construct is to take a single hexagonal cell to form a little row of these, and then you can form an array of hexagonal cells. Coalesce is where you don't stack an array. You actually let them drift into an arrangement that looks very similar to construct. Develop is where you let the cells change their shape in the array, and then they grow according to differential rules, and you end up with the same structure. And then generate is where you have a small number of cells, and the groups of cells replicate and form these fractal patterns. So this looks like a snowflake that emerges from this basic array here. So you'll use these in different contexts. You might use a construct and then a coalesce, or a develop and then a generate. And you can build different things from that. So yeah, I'll put this in the Slack if you're interested in reading more about this. So any questions at this point? So I don't know if Sushmanth is able to talk more, if he wanted to talk more. I know he was cut off earlier by technology.
Hi, Bradley. Sorry for the disturbance, actually.
Oh, that's fine.
I was kind of middle of traveling today.
Yeah.
I was getting back from my home to college, and last week I couldn't work much because my brother got a surgery to his hand. We both fell off from a bike, and it was kind of terrible. And right now we are working on the DevoWorm website only. We are hosting the whole publications which are written on synthetic biases were hosted here. And parallelly, we are working on the instance segmentation model. Actually, I was reading some related research papers about it. What is instance segmentation, etc., etc. While seeing, I was thought of implementing it in Chainer library, but I thought whole DevoWorm was on PyTorch, so I was thinking to implement in PyTorch only.
Okay.
And I want to mention some things. Actually, DevoLearn was working fine, but in DevoGraph stage 1, we are extracting the centroids to a CSV file. And for the DevoGraph stage 2, we need the volume of each cell and the centroids of each cell at different time frames, and even lineage tree is also needed. But these all can't be extracted from DevoLearn. Mainly the volume cannot be extracted. I watched some other video frames of DevoLearn segmentation also. It was not working properly actually.
Okay.
There is disturbance affecting these centroids. That is the reason I was thinking to propose this instance segmentation model. I want to explain it to Jiahang Li. That's the reason I am explaining all this. And whatever the work is I'm doing can be really helpful for DevoGraph only for the stage 1, because the whole graph embeddings in stage 2 are working perfectly fine. But creating a perfect pipeline to stage 1 and stage 2 this thing is needed, and the perfect centroids and volume should be needed. That's the reason I'm implementing this instance segmentation model.
Yeah.
And I was working on Hugging Face also. I want to host a model in Hugging Face as a transformer, as a DevoLearn as a transformer. I was working on it parallelly, but there are a lot of problems with it. There are a lot of things I need to learn to implement it. I was getting friendly. I was welcomed very friendly to the Hugging Face community. They are helping me actually. The guy named Radmo, he was helping me out to build a model hub in the Hugging Face.
Good.
I'm half through that, and that's what my update is. For the next few weeks, I will be working on instance segmentation model only. I'm implementing in PyTorch. I was trying to get the centroids perfectly, and I'm using a lot of stuff is going on. It would be really. I think I need to write a blog on this, and I need to share it with you. That's
Okay.
what is there. Okay, and that's what my update is.
Well, that's great. Yeah, thank you. Yeah, so that's great that you're able to get some support from Hugging Face, and that'd be great to have it as a transformer. And then also, yeah, the thing about the lineage information is that that's going to have to, I think, come separately. Although I think you can, you can easily get them out in C. elegans. You can easily get them out of the cells, so their position in the embryo basically tells you what cell identity they are, and then we have the information about lineage tree, so we can link those two together. And we've had GSOC projects in the past where we've done the sort of linkage. So I'm not sure if you're familiar with some of the stuff that was done, I think in 2017, 2018 in that era.
Actually, what the problem is in DevoGraph in stage 1, whichever CSV file we are extracting, that is the not same CSV file we are using in DevoGraph stage 2.
Oh, okay.
That's what I found out, because they are two different file names I saw in the code. So that is the reason I want to implement a new model. Actually, DevoLearn can't extract the volume of each cell also.
Okay.
But stage 2 needs the volume of each each cell. And the centroids is also needed for different time frames. For the model accuracy, we need exact time, centroids of each cell at different time frame. That's what I want to update. I want to clearly construct a DevoGraph stage 1 pipeline, which can be easily used through some commands. If we run that command, it just need to extract the centroids, and it need to be given to stage 2 of DevoGraph. And after that, topological data analysis could be done. Actually, a tool which you shared is very useful to me. TTTK toolkit. It is there in DevoWorm Slack channel actually.
Okay.
Through that, we can do the topological data analysis also. There is a separate library called we can give just microscopy image to it, and we can extract all topological data analysis from it, like depths of cells, birth rate of cells, and instance segmentation of those cells. We can cross verify our model and the topological data analysis toolkit output. So it's kind of a huge work. That's the reason I started working on it before the GSOC start. Whether I was selected or not, I don't need that, but I want to complete work and host it on DevoLearn.
Okay, yeah, that sounds great. Yeah. Let us know what you need for that. If there's anything that you need, like I know you were asking for blog posts. I gave you the Synthetic Daisies blog posts.
Yeah.
Those are just things that we have on our website that are useful to people. And then we'll probably do some other we'll probably use it also as, like I said before, like a documentation portal. So we'll have some other posts that will generate this summer probably on different things. And we'll have that all, up there in that spot. So someone can go there and read. what they need to.
Actually, when I came to DevoLearn, when I was starting to understand it, it was like quite hard to me. I mean, I can't understand, but the whole stuff is in different places. That's the reason Mayuk told me to build a blog for a whole DevoWorm, where if a person comes there, just see DevoLearn, particular blogs will be there. Digital Balenciaga. Actually, I contacted last year other guy also. I don't remember the name. He did GSOC from Orthogonal Research Lab. He's from my same college, Amrita.
Okay.
His name is last year GSOC. Two guys did actually on digital Balenciaga.
Oh,
One
Ojwal?
was not Ujjwal.
Oh.
Last year, two guys from Amrita College. One is Karan, and another guy is, I don't remember. I asked them to give some blogs or some documentation thing also. They are actually writing on it. When they did, we can host the whole blogs thing in DevoWorm website. And this could be useful for future also. Whenever someone comes and reads them, they will easily understand it and they can easily contribute to it.
Okay. Yeah. Yeah, I can't remember who. I can't remember who you're referring to right now.
I think the guy name is Karan and another guy name is Arvind. I don't remember.
Oh, right. Okay, that's a different project. Yeah, Karan and Hare Krishna. Right?
Hare Krishna, sorry.
Yeah, Hare Krishna.
I asked for them for a documentation actually last week. They are writing it and they will submit to me by next week. Then whole thing get pushed into the DevoWorm website for the blog section.
Yeah. Yeah, they did a different project last year. They did like a which we need to revisit too, which is the digital microspheres. So they built like a Susan actually has some data from a specialized microscope she's built, where she has like images coming from different angles, and then their job was to create like a sphere where you stitch those images together, and you can view, and kind of a viewer where you can spin the thing around and explore the surface. We haven't gotten past like kind of the basic viewer, so we've been trying to get it back off the ground, but it's been it's kind of hard when you don't have a devoted time to do it, and but that stuff has been there too. So I was thinking of putting that into DevoLearn as well, but it's kind of different in a lot of ways, so I don't know where to place it.
Actually, I was contacting every GSOC member who contribute to DevoWorm, and I'm asking for them to write some kind of documentation which will be useful. Actually, everyone responded pretty cool. Mayuk, Mainak, Ujjwal Singh. They are like writing the blogs and they are helping me. Actually, they are sending me. I got so many blogs. I need to create that Hugo framework. I need to push them all. Due to this lot of disturbance, I couldn't do the work. Maybe by next week or upcoming week, I will complete that and I will start working on instant segmentation model also for DevoGraph and topological data analysis also. And that's what my update is. Anyways, thanks, Bill.
Oh, no problem. Yeah, great. Sounds great. So yeah, thank you, Sushmanth. Let's see, some things in the chat. Okay, so Himanshu is I worked with Himanshu in our other group, and he did GSOC last year in the other group. He did a project on open source sustainability, so he did some reinforcement learning methods and built them into an agent-based model. So Himanshu said, I would like to introduce myself. I'm Himanshu Chougule, a third-year undergraduate pursuing CSIT from VGTI Mumbai. I've done my GSOC 22, I told you about that. I am interested in contributing to DevoWorm. Glad to be here. I'm currently studying the literature in topological deep learning. The recent paper shared by Jiahang, this archive paper, this was in the Slack that we have. If you go to the DevoLearn DevoGraph channel of the Slack, we have a number of things in there relevant to this summer's. We'll be doing this summer. So Jiahang shared this paper here. It's Architectures of Topological Deep Learning, a survey on topological neural networks. And this is on the archive. This talks about TDL, topological deep learning, and says, I think we just talked about this with respect to DevoGraph. So this is like the sort of like we talked about stage 1 and stage 2. Stage 1 being image segmentation. We've been working on that because that's kind of the fundamental aspect of it. If you don't have good data, it's hard to get stage 2 to work well. And then stage 2 is, of course, assembling the graph embeddings and creating graphs of the data. And then stage 3 is this topological aspect, topological data analysis. And so that's something we didn't really ever address. We just didn't have time, and it's very difficult to do that in addition to the other 2 stages. So Himanshu wants to do basically stage 3 of that. And so we need to kind of figure out, if you want to do that, you have to figure out how to do it. And so there's a lot of stuff on topological data analysis. This topological deep learning area is a good place to start, I think. Architectures of topological deep learning, a survey on topological neural networks. This was uploaded to the archive 4 days ago. This is a great resource. And then there's an associated GitHub repository called Awesome TNNs, which summarizes schemes in various topological domains. So this is the Awesome TNNs repository. Looks like they have, this accompanies this paper, Architectures of Topological Deep Learning. This talks about cellular complexes, combinatorial complexes, hypergraphs, simplical complexes, and then a bibliography. So I guess this is this is a message passing simplical network. I guess this is code for it or something, or at least mathematical. There's the mathematical notation. Okay, it was it's rendering now. So these are different schemes. Without getting too much into like what, because there's a whole sort of you have to like talk about the field, you have to define a bunch of terms. Without getting into that, you can go to that repository and kind of browse around and get a sense of the major areas that in this area. So like you have schemes in the hypergraph domain, you have hypersage, hyperattend. These are just different methods. Hyperconv, which is, I imagine, a convolutional network, all deep sets. So they're different schemes for this. And so, yeah, this is, I think, a good place to go and look and at least try to figure out what might work with what we're doing in DevoLearn. So, in DevoGraph. So, yeah, that sounds great. I look forward, talking about this more, Himanshu. And yeah, you might want to get in touch with Sushmanth and Jiahang. Jiahang did some of the original DevoGraph work last year, and then Sushmanth has been working on things this year with DevoLearn. And so we'll be getting more into this over the summer. Oh, the link to the article later. Yeah, I can all the articles I'll be sharing in the Slack. So that's good. Yeah, any questions before we move on? Okay. So this one is actually about, actually, like Susan has developed a good theme here in the meetings of talking about tensegrity and different tensegrity networks. So these are a little bit different than like the graph neural networks or the embryo networks we've talked about. This is where you have these networks of cells that have this structural integrity. They form, like we talked about in the synthetic morphology paper. You form these networks of cells in a morphology, and there are reasons why they exist for forming tissues, but also for physical integrity of the structure. So you want to have something that has, you can either control the stiffness or the passivity of the structure, or have it be hyper stable. And so one thing I found this weekend, I was looking around, and I had forgotten about this, but if anyone's familiar with the architect Antoni Gaudi, who did a lot of work in the early 20th century, and he built a number of pieces of architecture in Barcelona, in Spain, and he built like this huge church, this cathedral. It was kind of based on like an ant or termite mound, basically. It looked like a termite mound. It had this very odd architecture, at least in terms of what was being built at the time. And so he was a pioneer in a lot of bio-inspired architecture. And the reason I bring him up in this meeting is because he built these structures that were sort of that sort of described some of these biological structures. So a termite mound is known for its rigidity, but also for its ability to self-regulate. Termites will build these things collectively, and then they'll live there. And they have a lot of it's very highly they're able to regulate the chemical composition of their environments inside the mound. They're able to regulate the temperature. The design is such that it's both pretty stable. It has to be stable. It's always getting rain and wind and other animals pawing at it. But also you have these it's this collective structure, so it needs to be done without, like, I don't know if there's an architecture that they follow, but it has to be hyper stable with respect to the larger structure. And so there's not really any evidence that termites build things based on a master architecture. So they have this sort of self-organized collective way of building this. In any case, Gaudi came up with a number of different models for how these things may be built. One of the things he developed is this hanging chain model. And so this is parametric design of La Lettre. So this is the hanging chain model. This looks a lot like, this is actually a model of the church that he built in Barcelona, Colonia Güell, which is a church that has like this, looks like a termite mound or something that doesn't look like a typical European church. And so this is the sort of the skeleton of it. And if you look at this here at the top, it's a mirror image of this at the bottom. So what he's done is he's taken this idea of a hanging chain, or a set of hanging chains that hang from the ceiling, and he's turned it over, he's flipped it over and made it into a structure. So it is known that Gaudi preferred modeling architecture over drawing it, especially models made of chains hung from a ceiling where strings with small weights attached. Through experimentation with such models, he discovered a way to use traditional Catalan masonry techniques in new, more complex ways. A chain suspended simply from both its ends results in a catenary curve that naturally distributes the static load, in this case tension, evenly between the links of the chain. So this is what he's doing here. He's basically using a tensegrity principle here of using this chain to distribute the static load, which is tension, along the link, a set of links. When the shape is flipped vertically and the materials become brick or stone, then the static load, now compressive, which when it's flipped over, is similarly even distributed. It's evenly distributed. So when it's hung as a set of chains from the ceiling, it has a bunch of, it's able to manage tension. When it's flipped back over, when it's standing from the ground, it's dealing with compressive forces in the same way. What Gaudi did was apply this tension compression analogy to chains hanging from the chains, or arches superimposed on arches, asymmetrically, permitting him to design a much more fluid architecture. So he had these arches, then he had other arches on top of those, and so that sort of nested design was actually hyper stable, in fact. And so this is what it looks like inside the cathedral. He has these arches here, and then he has arches on top of arches, and the whole thing is stable with respect to the supports here. Sounds like we have something in the chat. Oh, cable net tensegrity. Yeah, Susan pointed out that's cable net tensegrity. So yeah, this is again, this is a wire-free model that they built in the museum here. And so this was all done without a computer. So this was all done in this sort of architectural context with wire models, and they built wire models to show kind of what this looks like. So this is another example here. This is from 1889, so you get a sense of what this looks like. And then this is the close-up view of, so using scale models made of chains or weighted strings, it was long known that an optimal arch follows an inverted Cantor curve. We talked about that. Yeah, and then there's some citations here, which are basically this is the thing we just read, and then this just kind of goes over some more of it. So that's it. I just found that interesting. Okay, Sushmanth had to leave. Well, thank you for attending, Sushmanth. Thank you for the update. Any questions at this point or comments you want to make? Susan, did you want to say something?
Not too much. I'm just curious as to whether you could have tensegrity structures for a neural net.
Oh.
Combine something to make, say, the network more flexible or self-supporting. I don't know.
Yeah. So, like in a lot of the graph neural network stuff, they're building neural networks that kind of are the topology of a graph. So, like the graph embeddings are just one way to do it. And people have actually designed the topologies where they're like, they sort of resemble the graph that they propose is underlying some process. So it stands to reason that you could build like a tensegrity neural network. You could have the weights be, something where they are, they translate into physical variables. So like the weights would be like the stability of the connection between two nodes. Like we have our strings and we have our nodes in a tensegrity network. Those would be, analogous to like two processing units and then like a weight. And then the weight would just vary with respect to the physical stability or the physical tension or compression that we want to give. So like the form finding tensegrity stuff that we talked about earlier this year, where we had those matrices where they described the strings between different nodes, and then those strings would have you'd have to find the stable structure, and the stable structure would be a description in the matrix of like stable stable links. What are the forces? What are the compression or tension components? And then finding the optimal one. And you could do that in a neural network, I think, just by translating those into weights and then saying, what is the optimal set of weights here?
Okay, that sounds like an interesting approach.
Yeah. So yeah, that's great. Yeah, anything else we want to say before we go? I think that's it for today.
I'm good.
Okay. And so yeah, I see a lot of turnout here for like the summer. We're starting the summer here. We're interested in these GSOC projects or working on DevoLearn and DevoGraph and related things. So yeah, let's keep in touch in the Slack on that. We'll be ramping that up soon. By May 4th is when they make their final selections. But if you're not selected for whatever reason, or if you didn't apply this year and you want to be involved, please feel free to participate. We can help you build things and learn about open source software and learn about how to contribute to open source projects. So there are a lot of benefits to being part of our group, and working on this. So if you have any questions, let me know about that. And then in the month of May, we'll be doing a community period with, it's related to something we have to do for GSOC, but we'll be doing this in the group where we talk more about like the organization. We'll talk about OpenWorm, the OpenWorm Foundation, and some of the things that they do and some of the resources they have. and then maybe some of the things we've done in the past here that are relevant to what people are doing now. Because we've done a lot of things in the group. We've been around since 2014. So we've done a lot of things that are sort of in the past that we don't revisit every week. So if people want to, find inspiration, there's probably a lot of inspiration in our group, things we've done in the past. We just, aren't talking about them all the time now. So we'll talk about those as well. And then we'll also talk about some, how to build open source projects in software. There are some readings that are really good, I think, for people to go through. I have a couple of books that I can share with you, and they basically go over a lot of the details of this. So this is a good opportunity to read about how to sort of build and market your own open source software, how to develop what it means to be open source and so forth. So that's all in the next few weeks. So okay, thank you for attending, and have a good week.
Okay, bye.
All right, bye.
Thank you. Bye.
Bye.
See you next week.
See you next week. Okay, now I want to talk about a few graph neural network papers that have been in either the Slack or things that we've been meaning to talk about for a while. Actually, this is graph neural networks, but this goes beyond graph neural networks to some other network issues that we might address or are interesting to our group. So the first paper here is Everything is Connected, Graph Neural Networks. Now this paper was put in our Slack channel in the DevoLearn DevoGraph channel, I believe. And so this is Peter Velikovic from DeepMind in the University of Cambridge. So this is on the archive, but it's also on our Slack. So this is the graphical abstract. It shows kind of what the idea of this paper is. So you have this representation here with inputs X and A. Then you have this GNN, which is this embedding that can be built from, so you have the inputs coming into X, then you have the latents coming into H. So this is the graph neural network structure, and many graph neural network structures that are generated from the same input data. Your latents are H and A, so your inputs are X and A, your latents are H and A. So the inputs get transformed to a set of latent states. And so that's where you get your graph neural network embeddings. Now, out of these different embeddings, we have three things here. We have node classification, which is Z equals a function of the latent space H. So H sub I is the nodes that we have in our latent representation, and we can classify those nodes accordingly. We have the graph classification, which is basically a collection of nodes forming a graph. And then we have link prediction, so we have links in that latent space that connect nodes. So this is just showing this the scheme here. So the paper starts out, in many ways, graphs are the main modality of data we receive from nature. This is due to the fact that most of the patterns we see, both in natural and artificial systems, are elegantly representable using the language of graph structures. So I don't necessarily know if I agree with this statement, that graphs are the main modality of data. You can certainly create graphs from anything, but as it holds true in network science, you have to have a natural sort of graph structure. That is, you have to have nodes and you have to have connections between the nodes. And those have to have some sort of discrete nature to them. So you can create a graph out of anything, but the best things that sort of are amenable to graphs are things that resemble this sort of discrete structure of highly interconnected components. So not everything in nature is like that. Sometimes other data structures are more suitable for those things. In any case, this term elegantly representable is interesting because they talk about in graph neural network literature about expressivity. And that means that a graph neural network should be able to express a lot of different, should be able to express representations for a lot of different types of systems, which is true. It's just not every system in nature. So prominent examples include molecules, representative graphs of atoms and bonds, social networks, and transportation networks. So I'll draw this on the board just to show an example of what we're talking about here. So a graph structure is basically where you have a number of nodes that are discrete things in the world. And these discrete things are connected by arcs or connections. They usually have some sort of quantitative value to them. So, we have our nodes here. And then we have like I, and then we could, say that J is this other node. And then there's a connection IJ, which is, this could have a strength, a quantitative strength. This could have just a regular value, usually at 0 to 1 in that interval. But it can be anything. Now in the meeting we've talked about tensegrity structures. And so in form finding tensegrity, we sometimes use matrices, which are actually sort of the representation of this. So this can be represented in a matrix. And then that matrix is, you can actually use that to find the best form. So in a tensegrity network, the matrix will have a bunch of entries in it. And then it'll describe the physics of the bonds between two things. So for example, if we have our nodes, which are like blocks, we have strings between the blocks, we introduce a tension here, and the value is here. And so form finding tensegrity is just finding the optimal matrix for hyperstability. So we're finding an optimal matrix. And this is form finding tensegrity. So I think in the meeting we talked about you might be able to like a neural network from tensegrity, but also graph neural network by extension, because that's what we're talking about here. So this is all sort of graphs look like this with a matrix, and then we can apply it specifically to like tensegrity or form finding exercise. Or sometimes, if you're dealing with a neural network, you already have that network structure and you already have the matrix, and all you need to do is you need to find like the weights, the optimal weights. And so graph neural networks really to feed into sort of more conventional neural networks. But it's important to remember that you need to have like a certain structure to your problem. So if you had a continuous problem, for example, where you had something like flowing water, graph neural networks might not be good for that. So there's a lot of potential for these kind of models. The main area of the short survey is to enable the reader to assimilate the key concepts in the area and position graph representation learning in a proper context with related fields. So what's interesting is that GNNs are kind of like phylogenetics in evolutionary biology. And so in evolutionary biology, phylogenetics is a field where we use directed graphs to find the true tree of life. And so we have a similar process. We take species, we take relatedness between species, we put it into a matrix, we optimize the matrix by certain rules, like what's the most efficient path through the tree for traits to be grouped together as having common ancestry. And then we have, we can do these searches on a number of trees that are generated and find the true tree. And so we do this with graph neural networks. We try to find the true graph neural network from a set of options that we generate based on our matrix. And so we actually have a precursor to this. And phylogenetics, of course, has been done for decades. So this is another sort of area that's sort of an allied area. So let me briefly go over this. So phylogenetics is where you have trees that look like this. You have some sort of common ancestor down at the bottom. You have, or I guess an origin. Then you have another common ancestor at the sort of the intersection of these two subtrees. These are usually called clades. And then your clades, of course, have species. They have tips of the trees, which are species, and then they have common ancestors within the clade. The idea is to have, like, say, a trait that appears in these two species. You say the common ancestry is here. This is more plausible than saying the common ancestry is here. And then, this is the common ancestry of this clade, which has this character and its outgroup, which is another clade. And then this is the common ancestor of the entire tree. So this should have the least, what they call derived, set of phenotypic characters. Now you could apply that to graph neural networks, in that you could look at the, you could look at the directedness of the graph neural network. So the arcs may have a directedness to them. They may have a bidirectionality, which would actually also be interesting. That's something that phylogenies don't have. And then, so you have that. You have directed, bidirectional. Sometimes they call phylogenies DAGs. That's a larger class, directed acyclic graphs, meaning that they go in one direction, they go this way, and they don't reticulate, meaning that these branches don't join back up. They just branch in their terminal. In a network, we have both cyclical and we have bidirectional. So I guess we could call them CBGs, if you want. I don't want to do that, but some people might. In any case, that's what we have. So, there are all different types of networks. We have molecular networks with chemical bonds. We have connectomes, which are neurons with synapses or other ways that they're connected. Graphs are universal language for describing living organisms. Truly, there are a lot of different ways, a lot of different networks and biological systems, in which I guess the point I'm trying to make here is that we can not only learn about graph neural networks from natural networks, but we also have to recognize that there are different types of networks within those natural systems. So we have the DAGs, we have the CBGs, we have the, just basically an all-for-all connectivity. We have a lot of different types of bipartite graphs. In our stuff that we do in embryo networks, we have these sort of composite structures, which are where they may be bipartite at one point in development, and then highly interconnected in another point in development. So there are all different ways you can model biological systems with networks. GNNs, this is a Wild West sort of different types of approaches, and I think like a lot of the model systems that they hang themselves on here, or that they mention, are only a subset, and I think they pick the easiest ones, the ones that are most similar to, say, a computer science problem. But that does not mean that you have to treat it like a computer science problem. You should treat it like a biological problem first, and then find the computational analog to that. Beyond this, it is likely that the very cognition processes, I don't know what that means, I guess cognitive processes, driving our reasoning and decision-making are, in some sense, graph structured. That is, paraphrasing a quote from Forrester, 1971, no one really imagines in their head that all the information known to them. Rather, they only imagine selected concepts and relationships between them and use those to represent the real system. So this, going back to the DAGs example, this is very much what you find with like sort of Bayesian graphs or Bayesian directed graphs. And people use those for decision making and other types of cognitive applications. So there are a lot of ways we can use GNNs and graphs, and there are a lot of intersections, both in biology and in cognition and in other domains. And then you talk about transformers, which is interesting, that actually transformers are themselves a special case of GNN. So I know there was some talk about turning DevoLearn into a transformer, and so that's something I think Sushmanth is working on. That is definitely going to lead us down the road to GNNs, so stay tuned for that. So there's some fundamentals. There's a lot of, you have to understand a lot of math. You have to understand permutation, equivariance, and invariance. So when we have graphs, when we have all those nodes and they're interconnected, we have to know that you can put them together in different ways and still get the same answer. So sometimes you can put them together in different ways. A problem can be solved with a number of different graphs. There's no way to say that one graph is better than the other. And indeed, in some of the work in phylogenetics, they've had cases where if you don't have enough data, you can have a bunch of equivalent trees. And so that doesn't necessarily help you narrow down your data set to the best tree, but that's what you're dealing with graphs. And with interconnected graphs or bidirectional graphs, it's even worse because there are a number of different sort of isomorphisms that you can have. So you can describe a certain set of relationships using many different types of graphs. This actually, though, is also helpful because sometimes those graphs, sometimes it winnows down your search space. So sometimes if you find a class of isomorphic graphs, you know that whole class of graphs basically solves your problem. And so maybe it doesn't matter which one you use as long as you use one of those. And so that cuts down your search space to some extent. So they go through invariance and equivariance. And then they go through, sort of graph neural networks itself. There's this issue of message passing, of diffusion and propagation. So the idea that a node will pass things to another node, or that there will be some communication to another node, or like I said, there's a directionality and bidirectionality. So if you have, let's draw this on the board again. So in directionality, we can assume that something passes from one node to the next node. And this is usually in time. So this is a flow of something. I'll use that. I'll use maybe like F, which is the flow, and then time. So this is a flow over time. This is a directional graph. So this is a directed case. This is the directed case. Right. So we have node A here, node B here. You have a flow from node A to node B. This happens over time. In the bidirectional case, we have two potential flows. They both happen over time, A and B. But what's interesting here is that you can have flows simultaneously in either direction. So in other words, in any given point in time, and this is the undirected case, in any given point in time, you can have something flowing from B to A or A to B. And so you can have a collection of things at any one time point. You can say T1 equals A to B, B to A, or just A to B in both cases. So, and you have a number of those events, so you can count them up. So it's almost like a distribution of flow over that period of time. And then you can get a density function, and you can, do interesting things with that. In any case, the point here is that you have these different approaches to passing on information. And so they call them spatial sort of propagation, spatial diffusion. It's basically moving across the graph, because if you move from A to B, that's just one step. But in a network, of course, you have a bunch of these So A might be connected to B, C, D, E, and F, and things diffuse from A out along these arcs. And so they may diffuse at different rates. They may diffuse at the same rate. They may diffuse bidirectionally at the same rate or a different rate. So all of these are possible. And so Bronstein, who's a big name in graph neural networks, talks about most of these events, these diffusion, propagation, or message passing events can be classified in one of the three spatial flavors. The first is convolutional, the second is attentional, and the third is message passing. And so this, these parameters will allow us to plug this into a neural network context, and it'll allow us to, account for that activity in the network. So this kind of goes over representative convolutional GNNs include the Chebyshev network, graph convolutional network, and the simplified graph convolution. Representative attentional GNNs include the mixture model CNN, graph attention network, and its recent V2 variant. And representative message passing GNNs include interaction networks, message passing neural networks, MPNNs, and graph networks, GNs. Okay, so given in such a GN layer, we may learn many interesting tasks over our graph by appropriately combining H sub U. I exemplify three principal such tasks grounded in biological examples. So these are biological examples. There's node classification, where you have a protein-protein interaction network, where you have this problem that's set up here. Graph classification, where we need to reduce all H sub U into a common representation. And this is classifying molecules from their quantum chemical properties, estimating pharmacological properties like toxicity or solubility, or virtual drug screening. So this is classification. So you want to take all of your H sub U, make them into a common representation, and then classify. Then we can predict links. So these links are, maybe assumed from the data. You might have a very clear definition of what a link is. In connectomes, we usually have some sort of physical link. We can measure that, and that's classified in, like, people have worked on connectomes where they've looked at microscopy data to look for gap junctions, or they've measured activity in the brain to measure, which units in your connectome are linked to which units. They could be, there's this idea of effective connectivity, and then there's anatomical connectivity. So those can be gotten from the data. But sometimes you want to predict the links. Sometimes you don't know what the links are until you actually have a prediction mechanism to say which things are most likely to interact. Or sometimes it's not clear from the data which things are most likely to interact over time. So you can actually build a model of link prediction. So we may be interested in predicting properties of edges, UV, or even predicting whether an edge exists, giving rise to the name link prediction. In this case, a classifier can be learned over the concatenation of features along with any given edge level features. So this is like, things like predicting the links between drugs and diseases for drug repurposing, predicting drugs and targets for binding affinity prediction, and predicting adverse side effects from polypharmacy. So you can use all of these different things as building blocks to build a graph. You can use graph classification, link prediction, node classification, and other techniques to sort of build a graph. You can build it from building blocks. You can build it from a real data set. Then you can also build GNNs without a graph, and that's where your transformers come in. Your transformers basically give you this optimal computation graph, which is where passing messages over it may be problematic due to bottlenecks, and so you want to optimize that network. And so you get an input graph, you want to optimize it, or you want to understand it better. And so it talks about transformers is GNNs. They aren't really a graph per se, but they're optimizing on a graph. Then there are geometric graphs, which go beyond permutation equivariance. We have assumed our graphs to be a discrete, unordered collection of nodes and edges. only susceptible to permutation symmetries. So in other words, if you change the order of how things are connected, you can find symmetries, you can find structures like simple complexes, you can find equivalencies in terms of their function. But sometimes you have systems that aren't discrete, you have more continuous systems. They may be, in fact, be amenable to graph analysis. Like I said, not everything is, but you would have this, you have this more continuous property, making it harder to actually extract what the real true graph should look like, especially over time. So if you're dealing with dynamical systems or dynamical contexts, extracting a graph like that operates over long periods of time might be very hard. So the graph is often endowed with some sort of spatial geometry. And I think if you look at the graph that I showed you here, this doesn't really have much of a spatial geometry. This graph, the phylogeny, has a very distinct spatial geometry. This little triangle has a spatial geometry. Sometimes the geometries can look very distinct. So sometimes if you have, there are things like the hairball network, which is where you have like a mass of links that are built around a mass of nodes. There's no structure there. Sometimes you have a circular network like this, where you get these nearest neighbor connections, and then you get sort of these connections that go around the circle. So there's this structure of, local and global connectivity. Sometimes the structure of the network is actually relevant to the geometry of the system. So like in a connectome, you might have something like this, and then something might go out to a tail or a head. So you have this anterior-posterior geometry, but you also have this left-right geometry. So, there are things like that that are very highly structured with respect to the geometry of the system, with respect to space. And so when we have a situation like that, it's actually both easier and harder to deal with. So we can exploit the spatial geometry to understand at least how things are connected. It gives the example of molecules. I did not, but that's a good example. Molecules as being structures that kind of look like the phenotype I showed you here, or not what we would call a hairball network. So hairballs are usually the thing that are very hard to understand, because this is both visually and of course using techniques like graph neural networks, because they don't really have a lot of internal and explicit structure. Something like this might. So this is the kind of thing we might be interested in designing a model that is equivariant, meaning it has equal conditions in different configurations, so not only to permutations, but also 3D rotations, translations, and reflections. So in this group, we talk a lot about embryos, and so one of the things about embryos is there's a lot of 3D rotation of the data of especially in development, where things fold, where things where cells migrate, and so your embryo networks will stretch, they'll flip in upon themselves, they'll connect. So you have like bipartite graphs, which are two distinct graphs which connect over time. And so all these things can happen, and these are things we really want to get at. So an E3 equivariant message passing layer transforms the coordinates and features and yields updated features f sub u and coordinates y sub u, or x prime sub u, x prime sub u. So these are the features and coordinates. So these are updates in time. So you have your features and coordinates, f sub u, x sub u, respectively, and then the prime, which is the update. There exist many GNN layers that obey EN equivariance, and one particularly elegant solution was proposed by Satoris in 2021, which gives the equations are given here. The key insight behind this model was that rotating, translating, or reflecting coordinates does not change their distances. So each set of nodes has a distance. So as you see here, especially if there's a geometry involved, this has a distance. These networks here have a distance here. You have distance 1, distance 2, distance 3. They're all distinct, And usually they have some relevance to the spatial organization or the function. So we can actually use that information to, we don't want to change the distances when we translate or rotate. And especially when we have an embryo network or a neural network in the brain, like a neural network, if those sort of those symmetries and fundamental distances are broken, then we can have functional deficits. So, a lot of this is sort of about mathematical transformation, but it's also about biological function. So operations where the distance is invariant, but the shape and the rotation and translation of the coordinate system, those are called isometries, and those are things we want to exploit and whenever we find them. So hence, if we roto-translate all nodes as x sub u, this equation here, which are x sub u plus b equals or leads to x sub u, but the output feature f sub u prime remain unchanged while the output coordinates transform accordingly. So we can have this output feature that's persistent, but the output coordinates transform. We look at the network in time 1 and time 2. It's changed its position, it's changed its rotation and orientation, but the output remains the same. So there are a lot of things we can learn from this sort of thing. So this is what they have to say about GNNs. There's some other GNN papers here. AlphaFold 2 is a GNN sort of, this is in this citation Jumper et al. 21. There are also protein-related papers which have made headlines in both Nature Methods and Nature Machine Intelligence. Mendez Lucio 21 and Gainza 20. Those are both papers that might be useful. Protein folding, protein design, and protein binding prediction all appear to be extremely potent area of attack for geometric GNNs. And so, one thing we might do is look at how structural biology relates to some of the stuff we're doing in development. So the next paper I want to talk about is architectures of topological deep learning, which we talked about in the meeting. This is also in the Slack. This is about topological data analysis and topological neural networks. So there are a lot of links to mathematics and physics here. And I'll just read the abstract on this paper. I won't get too deeply into it. The natural world is full of complex systems characterized by intricate relations between their components. Social interactions between individuals in a social network to electrostatic interactions between atoms in a protein. Topological deep learning, or TDL, provides a comprehensive framework to process and extract knowledge from data associated with these systems, such as predicting the social community to which an individual belongs in social networks, or predicting whether a protein can be a reasonable target for drug development, such as in some of the drug discovery networks. TDLs demonstrated theoretical and practical advantages that hold the promise for breaking ground in the applied sciences. However, the rapid growth of TDL literature has also led to a lack of unification in notation and language across GNN architectures, which is what they call topological neural networks. This is an interesting point because, in GNNs, it's bad enough. And I think we see in the last paper that there are these these areas of interest that people have identified, at least. But the language is, a little bit sloppy, depending on what kind of network you want to build, depending on what application you're using. You have message passing, you have other types of diffusion, you have other links to other types of models. So I think in both areas, but especially in this topological neural networks area, there's going to be an issue with language. And one of the problems with doing research, especially when you cross between biology and computational science, is that the language it's non-standard. There's going to be language that you use for biology and language that you use for computation, and it's going to confuse people from one area or another unless you define it up front. And so this is an activity I think that's useful, at least to some of what we're doing, is to really kind of define what we mean. Nail our terms down and make sure we understand, or make the audience understand what we're trying to do. So I think that's an interesting point. This presents a real obstacle for building upon existing networks and for deploying TNNs to a new real-world set of problems in the real world. To address this issue, we provide an accessible introduction to TDL and compare the recently published TNNs using unified mathematical and graphical notation. Through an intuitive and critical review of the emerging field, we extract valuable insights into current challenges and exciting opportunities for the future. So they kind of go through, this idea of relational structure, which we talked about with respect to graphs. These interactions are relational. So this node is related to this node in a certain way. It's either related as a direct connection or maybe an indirect connection or some of these other. So you can make multiple hops between this node and that node, or you can make a direct connection between them. And so those sort of relational structural aspects are really interesting, not only from the aspect of building networks, but I think in the past we've talked about relational biology and some of the applications to both relational biology and to category theory. So there's a lot of interesting stuff there under the surface. Once we build these sort of machine learning tools, we can then attack some of these other issues and kind of, we've already actually published a little bit on our thoughts on category theory in a paper in 2021, but we have we'd like to develop that further, but it's at a secondary priority. This is the structure of interactions between components in a system. In geometric deep learning, graph neural networks have demonstrated remarkable achievements in processing relational data using graphs, and we think of this as mathematical objects commonly used to encode pairwise relations. So these pairwise relations are the thing that we see in the matrices. So when we have matrices like this, these are pairwise comparisons. This is I, this is J. We get an IJ pair. So we get M sub IJ, that's the matrix. We have a collection of these, and we can build relational statements out of them. So this node and this node interact in some way, they're related in some way, and we can describe that using a number. So that's what we're doing. We're building relational structures based on pairwise relations. But the pairwise structure of graphs is limiting, of course. We can have many, we can have like three-way interactions, four-way interactions, etc. And in graph theory and in network science, people have been working on this. So people have been working on this idea that, you have these matrices that are just I sub j, Ij, M sub Ij, but you also have like three-way and four-way and five-way interactions that should be characterized. So people have done this with like separate matrices, other types of data structures that allow you to capture these. Topological deep learning leverages more general abstractions to process data with higher order relational structure. The theoretical guarantees of its models lead to the state-of-the-art performance of machine learning tasks. So this is something that we might explore a little bit more. I might actually come up with some interesting examples of three-way and four-way and five-way interactions, and maybe how these have been dealt with in network science. But they're very, suffice it to say, we're dealing with a complex system here. It's not just simply pairwise interactions, added together. There's a lot going on. This is a simple example in our hairballs, which we have here. There are many of these types of things. And so when you try to characterize like triangles and other types of simple complexes, which are just like shapes that we can pick out of the data, they don't necessarily mean anything. What they're just useful, categories that we might put in. So we might find all triangles in a graph, or in a hairball we might find all circles or circular paths. But that also means that there's this sort of higher order set of relations. Sometimes there are 3 and 4 and 5 nodes that are all interacting simultaneously. So to find those, would be useful for a number of biological problems. So that's, I think, if you want to read more about that, that's on the Slack. But it's also been pointed out in the meeting. This is a nice paper on accelerating network layouts using graph neural networks. So this is actually having to do with visualization. And so this is an application of graph neural networks. It's quite interesting. Albert Laszlo Barabasi, who is actually a pioneer in network science, is an author on this. It's nice. He's getting into graph neural networks. The abstract of this paper reads, graph layout algorithms used in network visualization represent the first and most widely used tool to unveil the inner structure and behavior of complex networks. Current network visualization software relies on the force-directed layout algorithm. And so if you use something like Gephi, you'll find the force-directed layout. It's just basically where you have certain options for like visualizing all these nodes and their connections. It kind of doesn't lay it out, typically doesn't lay it out in like a spatially explicit manner. It usually lays it out into where it finds space on the graph. So if you have a field of if you have like a figure that's a square, you might be able to plot something like a circular graph fine. But the circular graph is like the most sort of topologically pleasing shape relative to your display space. It doesn't necessarily represent like a biological it's not biologically explicit. It's just showing these connections in a way that you can see them and that makes sense to the viewer. So, that's not optimal. It traps large graphs into high energy configurations, resulting in hard to interpret hairball layouts. So the hairball layout like this isn't a very good image of it, but suffice it to say, you can't really find a lot of the structure just by looking at it because there's way too many connections and it's in this hairball because, again, it's the most efficient way to display it in this square box that we have here. But, you could get into it and explore it and maybe find other ways to do it, but that's not the way the force-directed layout algorithm works. So here we use graph neural networks to accelerate FDL, showing that deep learning can address both limitations of the algorithm. It offers a 10 to 100-fold improvement in speed while also yielding layouts, which are more informative. So again, having something maybe like this, which is anatomically explicit, would be better. But we don't often have the ability to do this using force-directed layouts. Part of this is because in very large networks, your search space is very large, and it's hard to find the optimal display. This goes back to this idea of finding the true tree in a phylogeny, right? You have millions and millions of trees sometimes to sort through, and you can't sort through all of them using current algorithms and current computational power. It's prohibitive. So you have to find heuristics. And the same holds true with displaying graphs. How do you find the absolute best layout? Well, it's hard to do. So we analytically derive the speedup offered by GNN, relating it to the number of outliers in the eigenspectrum of the adjacency matrix. So this is this matrix back here. The adjacency matrix is M sub ij, which is giving us these adjacency relationships. We get their eigenspectrum that you can extract from that, and you can look at different aspects of the graph that way, and you can actually relate the computational power of GNNs to this ability to sort of pick things out of the graph. Particularly, the GNNs are particularly effective for networks with communities and local regularities. So communities are just groups within the graph that are highly clustered together, and regularities are just, like the triangles that I mentioned, or different substructures in the network, like circular connectivity or something like that. Finally, we use GNN to generate a three-dimensional layout of the internet and introduce additional measures to assess the layout quality and its interpretability, exploring the algorithm's ability to separate communities and length distribution. So length is this like the length of the links in space. It's not the weight, it's the length, physical length. And then these community finding and community discovery have been big topics in network science. And so, there's been this quest sort of to find the optimal number of communities in the graph, or like the clusters of activity in a graph. And so it's hard, again, when you have this sort of hairball network, it's really hard to find communities just by eyeballing, because it's not like, actually, when you have a bivariate graph and you see clusters, those often can be misleading. You need a formal clustering algorithm to determine whether something is in one cluster versus another. Just eyeballing it isn't going to work. You can imagine taking that bivariate case and exploding it into this high-dimensional space. And you can see that it doesn't necessarily work to say that they're clusters. Even if things look clustered in a hairball, it's almost impossible to really define what nodes are part of that cluster and which aren't. So that's why we use these community detection algorithms. And so this actually helps with that task as well. The novel use of deep neural networks can help accelerate other network-based optimization problems as well, with applications for reaction diffusion systems and epidemics, or things that range from reaction diffusion systems to epidemics. So reaction diffusion systems are very familiar with that in this group, because that's one of the things we talk about when we talk about morphogenesis, that reaction diffusion systems can generate morphogenetic patterns and things like that. So it's really interesting that we have this sort of ability to use neural networks to do these things, graph neural networks especially. And so they kind of go through their method here for doing the graph layouts, but they also talk about some of the application domains as well. So, we have in this application, you have the number of nodes, you have the layout dimension, you have the weight and dimension, so you have a layout that's maybe desired, or types of layouts of the graph. But then you have these latents, which are there, which you can choose from if you want alternates. This shows the initial layout, which doesn't show very much structure at all. And this is actually a cube. So this is a highly connected cube, but we have to find this from the initial layout. This is a force-directed graph where it doesn't necessarily show the structure, it just shows the connections, and it displays it in a way that's kind of, fits it into this space. So it doesn't really consider any spatial information or anything else. And you can train it on this algorithm to make it sort of conform to the final layout here. So we can do this sort of rearrangement of the nodes in a way that's more informative. This kind of shows a similar thing here. This has comparisons between two different methods, I think, FDL and Newlay. So, this is FDL left column, Newlay 2 right column. So just saw that. And we have this interpretability of the layout. So now we have to interpret the layout in terms of statistics. So this is where you get again, like interpreting it by eye, which is a common activity in network science sometimes, to just see where things are, see the structure. Because sometimes when you do data analysis, you actually want to see things in sort of intuitively, instead of relying on statistics, which are actually more valid, but that's not the way people's minds work. So we want to be able to do this in a low dimensional space, and so you can see that this is where you're building these layouts that are highly interpretable. Quality, and this is an example of a hairball network here with some structure. So the hairballs are these masses of connections that aren't don't really have any structure, although this has structure along the edges. You have these clusters of hubs and nodes. Hubs are highly connected nodes, and nodes can be peripheral to the hub, but they can be in the same cluster. And so this is what they're showing here. And so this is the 3D layout of the internet. You're generating this hairball, but you're also generating some of the structure within the hairball. So you can see local communities. Sometimes it fails, the FDL fails to identify local communities. They actually have a video on Vimeo that shows the full 3D representation of this. So the 2D representation probably isn't enough to really see what's going on here. And so the proposed Newlay algorithm, which is what they've developed, is they show that it's superior to this FDL, which is the old approach, the force-directed layout. And they show that it's superior to that. They kind of, okay, that's about it. currently the efficiency of NULA is limited only by the computational complexity of GNN, which, while considerably faster than FDL, can be expensive or unexceptionally large graphs. So this is something you have to consider when you have very large graphs, is the computational costs. And we still have these run up against these problems, but especially with graphs, because you're dealing with these huge combinatorial problems. And so you have to really have maybe good sets of heuristics is the way forward, like we have with phylogenies, but maybe also just kind of having better algorithms. So this is, they don't really talk too much about the future directions here, but they do give some of the mathematics behind their methods. Finally, I'd like to talk about, just kind of briefly discuss this paper, make people aware of it. This is a paper where they look at unstructured protein aggregates. And so we've talked about aggregates having a role in development and having a role in self-assembly and self-organization of biological structures. So it's an up-and-coming topic talking about protein aggregates, especially like in vesicles or in cell cytoplasm. And so this is an interesting area. And in this case, they're building network Hamiltonian models. So they're actually applying networks to this biological problem. They're building this specific type of network, and it's a framework for sort of understanding protein-protein interactions, topologically complex protein-protein interactions. And so this is not graph neural networks, but it's an application where it's closely allied to it. So I'm just going to go over the abstract, and then we'll, I can make the paper available to people. We may talk about it in another video. This is from the Journal of Physical Chemistry. The abstract reads, network Hamiltonian models, or NHMs, are a framework for topological coarse grading of protein-protein interactions. So protein-protein interactions are pretty complex, and we don't necessarily want to simulate every aspect of it. But what we want is a coarse-grained representation. So it's like, topologically coarse-grained means we only really care about one mode of their interaction. So we take that mode, we select what it is, and then we build these networks. So we can do this, so in which each node corresponds to a protein, and the edges are drawn between nodes representing proteins that are non-covalently bound. So we take one aspect of their interactions and we use it as the computational model. Here, this framework is applied to aggregates of crystalline, a structural protein of the islands implicated in cataract disease. This is a functional protein. The NHMs in this study are generated from atomistic simulations of equilibrium distributions of wild type, meaning the non-mutated genotype, and the cataract-causing variant W42R in solution. So there's a mutational variant W42R that's cataract-causing, and we want to know how that is distinguished from the wild type, or people without this variant. Or I guess eyes. I don't know if it's in humans or not. So this kind of talks about this. Network models are shown to successfully reproduce the aggregate size and structure observed in the atomistic simulation. So you're looking at these crystalline structures that form as cataracts, and you want to sort of understand the size and shape of them. You want to simulate that and reproduce it. So these crystalline proteins coalesce, they aggregate into these things that block your vision or that sit in your retina and do bad things, non-desirable things. So network models are able to successfully reproduce the aggregate size and structure that are shown in simulation, because they can't necessarily access this process in an experimental setting necessarily, so they want to simulate it, but they also want to use this network model and then provide information about the transient protein-protein interactions therein. So they just really want to understand the interactions with this network model. The system size is scaled from the original 375 monomers to a system of 10,000 monomers, revealing a lowering of the upper tail of the aggregate size distribution of the variant. Extrapolation to higher and lower concentrations is also performed. So you can do these titrations where you change the concentration and see what the simulation and the interaction should look like. These results provide an example of the utility of NHMs for coarse-grained simulations of protein systems, as well as their ability to scale large system sizes and high concentrations, reducing computational costs while retaining topological information about the system. So Hamiltonians are these dynamical systems models, and you're just simply treating each node as a Hamiltonian that evolves over time, and you're connecting them in a network. So that's basically what they're doing here. I don't know if there are any good images here. This just shows an example of the aggregation graph, where you have these monomers that aggregate, and they form the structure where they're interacting in certain ways. So it looks like when they come together, they have this linkage in the network. So some of these are linked together physically, but then you can see this in the graph. So you have basically this circle, the circular sample with a bunch of dots, which are these aggregates, and then the aggregates bind together. And so then they form a link, and then you have these little, subgraphs, which then maybe connect, and then you have a cataract. And so in the mutant, it's much more likely that they connect and interact in this way. So this is basically a highly spatially explicit graph, and so we're observing the graph microstate. We have this language, we're using a Hamiltonian in this. Given a proposed set of model terms, choice of time interval, we perform parametric inference using the pooled maximum likelihood method of GNN butts, which we can then infer this parameter using the relations of equation 3, which is here. As our goal is here to reproduce the distribution of aggregate sizes corresponding to component sizes. So each of these little subgraphs is a component, and then they link together, and there's a concept called the giant component, which is from deep in graph theory history, which describes this sort of network that spans the entirety of the system. And so the giant component doesn't, it's fully connected. You can go from one end of the network to the other walking across. So that, or, you can do a random walk across it. And the size is dependent on the shape of the network and the properties. So that's what they're trying to do. They're trying to find these components of different sizes. Maybe they'll find a giant component if it spans the entire network. But that's what they're getting at. They're using Hamiltonians. The term in H reflect multi-body interactions is reflected in the topological degrees of freedom of the aggregation graph. So yeah, and I guess people have used this before in different contexts. But this is kind of a taste of the network Hamiltonian and how people use networks in these kind of systems. And some of these issues that we talked about with respect to GNNs and simulations and dynamical systems and all that. So as you can see, there's a lot of potential for GNNs, and we can talk more about that in another meeting. Hope you found this useful, and I hope you learned something.